All right. Now the next concept we're going to talk about here is called the transport maximum. The transport maximum. And we mentioned earlier that glucose moves, or glucose, all of the glucose is reabsorbed in the proximal convoluted tubule. All right, I want you to stick that in the back of your head. There should be no glucose anywhere else along the tubule. All of the glucose, 100%, is reabsorbed in the proximal tubules. And we'll talk about the mechanism in a minute. So what we want to understand here is that this requires a glucose transporter. We talked about these last semester. A glucose transporter is a protein sitting in the membrane that's transporting glucose into the cell, right? And so these proteins are present in a finite amount, meaning that there are only a certain amount of these carriers. They also have a finite rate, meaning that they can only operate at a certain rate. Okay, think about a taxi stand or a bus stop, I don't know. You have a certain amount of taxis and they can only carry a certain amount of passengers. And so there's going to be a certain passenger number where you are maxed out. You cannot carry anyone else because of that finite amount of carriers and their finite rate. They can only go so fast. And so this is the case with glucose and the proximal convoluted tubule. There's a set amount of those glucose transporters, and if we exhaust them, we can no longer reabsorb glucose. What do you think is going to happen? What do you think is going to happen here? If we can no longer reabsorb glucose, where will we see it? In the urine. It's going to spill into the urine, where it should not be. This is what happens with diabetics. They've maxed out all their glucose transporters, and so we have glucose spilling into the urine. Now let's talk a little bit about how glucose is reabsorbed. So on the apical membrane, there's a secondary active transport. Someone remind us what secondary active transport is. Again, concepts from last semester. What is secondary active transport? So we know it's active, meaning it requires energy, right? Because it's secondary, does it require ATP, or is it going to hitch a ride on someone else's energy? It's going to hitch a ride, right? It's going to hitch a ride. Secondary active transport means I'm going to use one molecule, let it move down its concentration gradient, that's going to create some energy, and I'm going to borrow that energy. Okay, so that's secondary active transport. And so that's how glucose is reabsorbed at the apical surface. That's how we get it into the cell. How is it absorbed at the basolateral membrane? So across from the cell, right, that epithelial cell of the tubule into the blood vessel. This is where we have the carrier. This is where we have those GLUT4 transporters, and that's an example of facilitated diffusion. Okay, so on the basolateral membrane, we have the regulation of the movement of glucose by the presence or absence of these GLUT transporters. Now let's look at an example of a transport maximum here when we talk about how the kidneys filter out that glucose. So your plasma glucose level is typically about 100 milligrams per deciliter. That can change if you're in the fed state or if you're in the fasted state. That can vary, but not very much. Okay. Our filtered glucose load is about 125 milligrams per minute. Someone remind us what the filtered load is. What is the filtered load? We talked about this yesterday. So the filtered load is how much of this molecule, how much of this substance is filtered per minute. Okay, and for glucose, it's 125 milligrams per minute. This gives us a transport maximum for glucose of 375 milligrams per minute. What does that mean? What does that transport maximum mean? What does that 375 mean in your own words?
An finite amount of 375 milligrams of glucose can be reabsorbed.
Right, so 375 milligrams per minute is the maximum amount of glucose that my transporters can handle. After this 375 milligrams per minute, I'm going to start to see glucose in the urine, right? I've hit my transport maximum. Now, that figure, that 375 milligrams per minute, correlates to what we call the renal threshold. And because there's a little bit of discrepancy between the scientists, the biochemists, and the clinicians, there are two values for this renal threshold. There's a theoretical value, which is 300 milligrams per deciliter, and then there's an actual renal threshold. The theoretical value is what was deduced based upon studying the cell exclusively, so not studying it in the context of human physiology, but just looking specifically at this one carrier on this one cell. And they came up with a number of 300 milligrams per deciliter. Now that is the blood glucose level, okay? And so that number means when my blood glucose is at 300 milligrams per deciliter, I will start to spill glucose into the urine. That correlates to that 375 transport maximum. Clinicians, on the other hand, took the entire extent of human physiology and actually did clinical studies and looked at patients and figured out what was really going on. And they came up with an entirely different number. You can see a huge discrepancy here. Right? They got 160 to 180. And that was sort of, this process is what guides our daily intake for glucose. So our glucose should not go above 160 to 180 because of this reason. Again, above this threshold, above this 180 milligrams per deciliter, we'll start to see glucose in the urine. Okay?
So we need to check our blood sugar right after eating a big meal, which should not reach 160.
It shouldn't exceed 180. It certainly shouldn't. But if you look at the filtered load, so the filtered load, let's say I have a blood glucose of 180, my filtered load is only 225. So I have sort of a buffer zone between that 225 and 375. So I should still be theoretically safe, right, to have a blood glucose of 180. It's above 180 that things sort of get into that trouble area. Okay, so think about the filtered load, and the filtered load is 225. What's our transport maximum? 375. Okay, so even at a high glucose of 180, which is really high, I should still be okay to not see protein in the urine, glucose in the urine. Alrighty? Any questions on this concept, transport maximum? Any questions? Now again, I won't hold you to memorizing any of these figures, but just have a good understanding of the concept and be able to define the words in your own, or the terms in your own words. So transport maximum, what does that mean? The renal threshold, what does that mean? Et cetera, et cetera. The filtered load, what does that mean? And this table sort of ties this nicely together. So our transport maximum is about 375, right? That's this dotted blue line. And if we look at three processes, so filtration, reabsorption, and excretion, in the context of this transport maximum, we see that filtration rises steeply. Filtration never stops for glucose. Why? Is anything stopping it in the glomerulus? No, because it's going to keep going right through. So the limiting factor here is how much can be reabsorbed. So reabsorption at the transport maximum levels off. Right, we stop reabsorbing at this maximum. And so we can see this graph take a nice turn here and level off. All right, we can also see that excretion starts to rise and steeply does so after the transport maximum. And so that tells us that after that, you hit that transport maximum, filtration continues because that is not a factor that you can stop. We're going to keep filtering glucose regardless. The absorption stops abruptly and then excretion continues to rise, meaning that we'll spill that glucose into the urine and we'll see it in urine. And this is an old way of how scientists would test for diabetes. They would actually taste the urine, and if it was sweet, you would know that glucose was in the urine, and that was a hint right away that you're spilling glucose. And again, you should have no reason to have glucose in your urine except that your blood sugars are really high and there's some sort of pathology. That's why they call diabetes mellitus mellitus. The word mellitus actually means sweet versus insipidus, which means watery. And so that's because of the tasting of the urine which happened. Let's move on to secretion. Now, secretion we mentioned is the same process as reabsorption, but in the opposite direction. Right? The barriers are going to be the exact same. We have the basement membrane of the epithelial cell. We have the endothelial cell on the papillary side, and then we have the cell itself, right? The cell itself. So those three barriers remain the same. What differs is the direction of the movement. So substances are moving here from the blood into the tubule. We're getting rid of things that we actually realize we don't need, we can get rid of. For the purpose of this class, we're really going to focus in on potassium and hydrogen. Hydrogen becomes very important with renal balance, right? Acid-base balance. Other things here are secreted, such as penicillin, any of the drugs or medication that are cleared through the kidneys. Some drugs are cleared through the liver, so not all drugs. Creatinine, choline, but we're not going to focus on that. Potassium and hydrogen are the ones we are interested in. Okay. In the interest of time, I'm going to skip over these, but we will come back to them in our review. So let's talk about the regional specialization of the tubule. The regional specialization of the tubule. And all that this means is what is different, what is unique, what is distinguishing about each area of the collecting tubule? How does the proximal convoluted tubule differ from the distal convoluted tubule? How does it differ from the loop of Henle, the ascending and descending limb? How do all of those structures differ from the collecting ducts? And that's what we're going to sort of go through at this point. So again, I keep hammering this in here, but we have unregulated or non-regulated reabsorption taking place in the proximal tubule, as well as the loop of Henle. Okay, the regulated reabsorption is going to happen in DCT, which is the distal convoluted tubule, and the collecting duct. So keep those two distinctions very clear in your mind. Also, we want to talk about the function of water conservation. So the loop of Henle is very important in water conservation. In fact, this is an evolutionary adaptation. Our ability to concentrate the urine is directly reflected by the length of our loop of Henle. Animals in the desert, such as desert rats, kangaroos, have a very long loop of Henle, and this allows them to conserve their maximum amount of water. Okay, and they pee almost nothing. Whereas animals that are not adapted for the desert have very short loops of Henle. Okay, and they pee a lot. And again, this is an evolutionary adaptation based upon access to water. Okay, which again is very clear that's going to be limited in a desert environment. And we'll keep revisiting that concept as we talk about the loop of Henle. All right, on this table from your book, there's some substances, exactly where they're reabsorbed. The take-home ones that I want you to focus on is the proximal convoluted tubule reabsorbing all of the glucose, all of the amino acids, 85% of the water, sodium, and chloride. The loop of Henle, we're only going to focus on water there, right? The reabsorption of water. So between the PCT and the loop of Henle, we have most of the water reabsorption. That is unregulated or regulated? Unregulated, good. The distal tubule, the distal convoluted tubule, and the collecting duct, these are regulated, heavily regulated, meaning that based upon your hydration status, based upon the neuroendocrine system, based upon whether you're bleeding out or you're dehydrated, water may or may not move, right? It doesn't just move the way it does for the PCT and for the loop of Henle. Alrighty? Any questions so far? Any questions? Okay. So this slide again kind of just sort of reiterates that same concept we've been talking about. The proximal convoluted tubule, most of the water, sodium, all of the glucose. We have the brush border, which is formed by what structure? The brush border? Microvilli, right? So the brush border by microvilli. But then we also have this other interesting adaptation here. Leaky tight junctions. Anyone remember what a tight junction is? Get another concept from last semester. What is a tight junction? What is its function? So contrary to the name junction, a tight junction is the area between two cells that prevents paracellular movement, or that prevents paracellular movement, typically. But because we have leaky tight junctions here, we do have this paracellular movement. And so think about this being the tubule. Think about this being the lumen of the tubule, this being the basolateral membrane, which is closely associated with the capillaries. And so in between these cells, we should typically have tight junctions that are well anchored to each other, preventing the movement or the leaking of substances out into the blood. But in the case of the proximal tubule, because this is so unregulated, this leaky tight junction actually allows for transport of substances. And so this is the only place along the entire tubule where we see leaky tight junction. So I want you to stick these three unique identifying properties of the PCT in your mind: the leaky tight junctions, the brush border, right, and the association with the peritubular capillaries. Okay, so the leaky tight junctions, the brush border formed by microvilli, and the association with those peritubular capillaries. All right, so this again illustrates that. Let's move on to regulated reabsorption. Regulated reabsorption. Now here again, I keep mentioning regulated means that external factors come into play. External factors are going to be at play here. Whether or not this person is well hydrated, whether or not they have ADH in their blood, whether or not they're hemorrhaging, depending on their hydration status, depending on their neuroendocrine status, is their fight and flight system engaged of the autonomic nervous system, is their parasympathetic system engaged, the rest to digest. So these or this area of the tubule is under the influence of the hydration status, external factors of the body, let's say that, right? So external factors. And so what we see here is very tight junctions. So these are not leaky at all. Why would we not want these to be leaky tight junctions? How does that facilitate the regulation? Why is it important that we have extremely efficient tight junctions here? Why are we avoiding paracellular transport in this area of the tubule? Because this is regulated, all right? This doesn't just happen. This is at the mercy of external factors. And so you don't want to just have leaky tight junctions. Let's say you were dehydrated. Would you still want water to just be leaking across this area? No. And so because this area of the tubule is highly regulated, we don't want any leaking, we don't want any paracellular movement the way we would allow for the PCT. Okay, it's a very important connection to make. Okay, so again, this image is for just for you to let that sink in, make notes, etc. All right, touching again on water conservation. So the loop of Henle establishes conditions necessary to concentrate the urine. We're going to talk a lot about this coming up between today and tomorrow. We'll talk about the medullary osmotic gradient, which means that there's a difference in the concentration gradient. There's an environment that's set up in the medulla and the cortex to facilitate water reabsorption. So the loops of Henle can be equated with that part of the kidney that is really, really focused on keeping water in the body. And so the longer the loop of Henle, I said this before and I'll say it again, the longer the loop of Henle, the more or the better, the more efficient the ability to concentrate the urine, to conserve the urine, to minimize the amount of urine, to keep water. The shorter the loop of Henle, right, the less efficient at that process. And so you're just going to pee or eliminate water. You lose lots of water, and you don't have that ability to conserve and to consolidate urine. Okay. So we want to make the connection between the length of the loop of Henle and the ability to concentrate and conserve urine. Now, we're not going to talk about excretion rate. We're not going to talk about clearance. So you can X those slides, and we'll skip right down to micturition. All right, and this is something we touched on yesterday, so we'll touch on it slightly again, and then we'll start with packet 2. So micturition, again, is a fancy word for urination or peeing, and We know that the bladder is responsible for micturition. We want to really remind ourselves that urine is made in the kidneys. Okay, a lot of students may have that misconception: urine is made in the bladder, made in the kidneys, stored in the bladder. How does it get from the kidneys to the bladder? Ureters, right? So bilateral ureters coming from each kidney, insert at the trigone, right, by the ureteral orifices into the bladder. And so we store urine here until it's time to be voided. What was the name of those two sphincters we talked about which control bladder function? Talked about two of them, external and internal urethral sphincters. Which one was under our voluntary control? External, correct. What was the filling volume at which we lost that external control? 800 milliliters, that's correct. And so we want to remind ourselves here that although we have voluntary control over the external urethral sphincter, there is a volume where we lose that control. After 800 milliliters, the bladder must void. And so that the external sphincter will open and you will pee, okay?